Hello to the grade 9 students of Taft National High School. I am Sir Peter, a science teacher, and I have made this video to guide our students in answering their science learning activity sheets. For week 3, the topic is about heredity, inheritance, and variation. When we talk about heredity, this is the sum of all biological processes by which characteristics or traits are passed on or transmitted from parents to the offsprings. So we'll be dealing on how traits are acquired by the offsprings coming from their parents. The father of genetics is Gregor Mendel, and he has this Mendelian pattern of inheritance in which, according to him, the effects of the recessive gene are not observed when the dominant gene is present. So on this topic, we have to follow that there is dominant and recessive gene. However, there is this pattern of inheritance which do not obey the Mendelian pattern of inheritance, and this is non-Mendelian pattern of inheritance, which is our topic for week 3. Number 1 is incomplete dominance, second is codominance, and lastly is multiple alleles. Before we proceed to these topics, I will be presenting to you the words or the terms which you will be encountering for the duration of these learning activity sheets for week 3. Number 1 is genotype, another one is phenotype, we have allele, we also have homozygous, heterozygous, and lastly, the Punnett square. So what is an allele? So an allele is a variant form of a gene, and it is being represented by a letter coming from the parent, for example. And another one is genotype, in which it is the organism's complete set of genes. This is now composed of 2 alleles inherited for a particular gene. If an allele is a single letter, now a genotype is composed of 2 alleles. So meaning, it is composed of 2 letters or more. For phenotype, this is now the observable physical properties of one organism. The one in which we can see, we can observe, the color, for example, the length, the size, the form, and so on. So for heterozygous, it refers to having 2 different alleles of a particular gene. So if the letters representing the allele is different, meaning that particular trait or gene is heterozygous. Another one is homozygous, from the prefix homo meaning same. It has 2 identical alleles of a particular gene. So you have to look into the letters or the genotypes or the alleles. If they are the same, then they are considered as homozygous. Next is the Punnett square. The Punnett square is a graphical representation of the possible genotypes of an offspring arising from a particular cross. You will observe and you will see, if you have listened to your grade 8 science, the Punnett square was introduced there for the 4th quarter about patterns of inheritance also. So it's actually a 2x2 table in which the parents, the mother and the father, are placed at the left side and at the top of the Punnett square, and what is placed inside or indicated inside are the genotypes and the phenotypes of the offsprings. Okay, so let's start for the non-Mendelian pattern of inheritance. First one, we have the incomplete dominance. It is a form of intermediate inheritance in which one allele for a specific trait is not completely dominant over the other allele. If the definition of incomplete dominance is quite complicated, well, consider yourself mixing a white paint and a red paint. If you're going to mix them, what do you think is the product or the color when you mix the red and the white paint? Is it red? Is it still white? Or it will produce a different color? Now, if your answer is pink, well, you're correct. If we're going to mix white and red with the same amount. So, more or less, incomplete dominance is like that. The parents, for example, the white paint and the red paint, they are the parents. However, the offspring now is not red, is not white. However, the characteristic is intermediate. That's why it's called incomplete dominance. It is not, one particular trait is not dominant over the other. That's why it is incomplete. So if you're going to look into the table for genotype and phenotypes of 4 o'clock plant, you will see there the heterozygous and homozygous alleles. So for the RR, that is the genotype, and the phenotype is red. For WW genotype, the phenotype is white. Take note that RR, they are the same, meaning they are homozygous, and WW is still the same, meaning it's still homozygous. And the RW genotype is now heterozygous. That is correct. So its phenotype is now pink. So let's take into consideration the example that was given in your learning activity sheet number 9. Example 1 there. A pure red flowered 4 o'clock plant is crossed with a pure white flower 4 o'clock plant. So the two parents are red flower and a white flower. Okay, so red flower, what is its genotype? If your answer is RR, then you are correct. What is the genotype of the white flower 4 o'clock plant? If your answer is WW, then you are correct. Then how are we going to do that? How are we going to show the possible offsprings using the Punnett square? The first thing that you are going to do is to identify the genotypes of your parents. So the parents are the red flower and the white flower. The genotype of the red flower is correct, it's RR. And the genotype of the white flower is WW. The next thing that you are going to do is to make your Punnett square. And you will now decide as to where you're going to place your parents, the genotype of your parents. So if you decide to write the genotype of the red flower on the left side, it's okay. And if you're going to decide to write the red flower on the top part, it's still okay. Okay, let us write the genotype of the red flower on the left side. And the genotype of the white flower at the top of your Punnett square. The next thing that you are going to do is to cross the genotypes of the parents and arrange it alphabetically. Okay, so you copy R, you copy W. You're just going to do with the rest of the tables or to the boxes. Copy R, copy W, copy R, then W, R, lastly W. So you have now the genotypes of the offsprings of the red flower and the white 4 o'clock flower. So the red parent, it is homozygous. The white parent is homozygous. However, the offsprings of the red and the white 4 o'clock plant is heterozygous. We have RW, 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 and RW. So you now identify the phenotypes of the heterozygous 4 o'clock plant, which is pink, correct? Okay, so you are done solving the Punnett square of the offsprings of a red and a white 4 o'clock plant. For your learning activity sheet number 10, it's about the non-Mendelian pattern of inheritance, codominance. Codominance, it is in which both alleles are expressed equally in the phenotype of the heterozygote. Consider yourself mixing a jar of green marbles and red marbles. After mixing them, what is now the color? If your answer is still both colors will be present, then you are correct. Red and green will still appear after mixing the green and red marbles. more or less codominance is something like that. Let us check table 1, which is about genotype and phenotype of the bulls, cows, or cattle. So for the genotype RR, the phenotype is red. For the genotype WW, the phenotype is white. So again, look into the homozygous genotype. We have RR and WW. The letters are the same, so they are homozygous. Now let us look into the heterozygous, which is RW. If you will notice that there is a mixture of red blotches and white fur of the cow, that is now the roan cow or roan bull. So the genotype is RW and the phenotype is roan. Let us have an example. A red cow is crossed with a white cow. Now for codominance, in using the Punnett square, you still have to follow the same steps that I have done in doing or in solving the problem in incomplete dominance. So first, identify the genotypes of the parents. So we have red cow and white cow. The genotype of the red cow is RR, and the genotype of the white cow is, again, you're now going to make your Punnett square. And you are going to decide where to place the genotypes of the parents. If you're going to place the genotype of the red cow on the left side, it's okay. And if you're going to place the genotype of the red cow at the top, it is still okay. So you decide. Let us write the genotype of the red cow on the left side and the genotype of the white cow at the top. The next thing that you are going to do is to cross the genotypes of the parents and arrange it again alphabetically. Copy R, copy W, R, and W. R, W, R, and W. So you now have the genotypes of the offsprings when you cross a red cow and a white cow. So you now identify the phenotypes of the offsprings. So RW in codominance is not pink. Instead, it is roan. So you will see a cow with white fur and red blotches. There you have it. So you already have a Punnett square showing the offsprings of a red cow and a white cow. The last non-Mendelian pattern of inheritance is multiple alleles. It happens when a particular gene that exists in 3 or more allelic forms. So we have blood types as an example, the ABO blood type system. Now, there are 2 antigens that determine the ABO blood type system. The presence of these 2 antigens identifies the phenotypes of the blood types. So, for blood type A, there is the A antigen. For blood type B, there is the B antigen. For the blood type AB, there is both the presence of the A and the B antigen. However, if there is absence of both antigens now, this is now called the blood type O. So let us look into table 2, the human ABO blood types, the phenotypes and their genotypes. If you will observe, for blood type A, there are 2 possible genotypes. For blood type AB, there are 2 possible genotypes. However, for AB and O, there is only a particular genotype. So for blood type A, there is homozygous and heterozygous. So for the first genotype, it is homozygous because the alleles are the same. However, for the second genotype of blood type A, you can see that there is a different allele for heterozygous A. Capital I, A, and then small I. Okay, for blood type B, you also have two possible genotypes, the homozygous and heterozygous. For homozygous blood type B, it is stated there IB, IB. For heterozygous blood type B, you have their IB and small letter I. For blood type AB, you have their IA, IB. And for blood type O, because it does not code for an antigen, it is represented by two small letter I's. That is now that genotype. Let us have an example. A blood type of a father is AB and the mother is heterozygous A. What are the possible blood types of their child if the mother gives birth to their firstborn? Example 1. The blood type of a father is AB. So its genotype is IA-IB. And the mother is heterozygous A. So the genotype is IA small letter I. The question is, what are the possible blood types of their child if the mother gives birth to their firstborn? In solving problem sets in multiple alleles, you still have to follow the steps that I have followed in doing or in solving the Punnett square in incomplete dominance and codominance. So step 1 is to identify the genotypes of the parents. So for sample problem number 1, the blood type of the father is type AB. Blood type AB. And looking into the table that I have provided you, identify the genotype of blood type AB. Please take note that the letters A and B on the genotype are exponent or superscript. And for the blood type of the mother, it is heterozygous A. Heterozygous A, meaning the alleles are different. So, your genotype is IA and small letter I. The next step is to make your Punnett square. And you're going to decide where to put the genotype of the father and the mother. Okay, let us put the genotype of the father at the left side. IA, IB, IA, and small letter I. So, next step is to cross again. And again, arrange it alphabetically. IA, 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 I, IB, and IA. But take note, you have to arrange it alphabetically. So you include first IA, and then IB, and then IB, and small letter I. So you now have the possible genotypes of the offsprings when you cross a blood type AB father and a heterozygous A mother. But your task is not yet done because you still have to include your phenotypes of offsprings. So IAIA, its phenotype is homozygous A, homo A. IAI is just like the father, right? I mean the mother. So it is hetero A. IAIB is just like the genotype or the blood type of the father. That is blood type AB. And lastly, another one is IBI and it is heterozygous. So it is hetero B. Okay, so you now have the possible phenotypes and genotypes of the offsprings of a cross between a blood type AB father and a heterozygous A mother. The next thing that you have to remember is that each box on the Punnett square corresponds to 25%. So this is 25%, 25, 25, and another 25% to make it 100%. So if you're going to ask about the phenotypic and the genotypic ratio, or the chances or the possibility of obtaining a blood type, you have to remember that each box corresponds to 25%. So for example, what is the chance that the offspring will be blood type A? Homozygous. Homo, hetero, hetero. And this is only blood type A. So one box corresponds to 25%. So the answer to the question, what is the chance that the offspring will be blood type A, homozygous A, the answer is 25%. And the next, if the question is, what is the chance that the offspring will be blood type AB? So you look into the blood type AB again. Blood type A, blood type A, blood type B, blood type AB. So again, this box corresponds to 25%. So that is 25% chance of obtaining blood type AB if you cross a father with blood type AB and a mother with blood type A heterozygous. Now, for the phenotypic ratio and genotypic ratio. Genotypic ratio. You look into the genotype. IA, that is 1. Is there a genotype which is the same with IA? We cannot find. So 1. How about IAI? Is there the same? None. So it's only 1. Another one. IAIB. There's only 1. So the answer is 1. Lastly, IBI. There's only 1 IBI. So your genotypic ratio is 1:1:1:1. For your phenotypic ratio, you look into again the phenotypes. Homozygous A, do you have the same? There is none. 1. Heterozygous A, there's only 1. Blood type AB, there's only 1. And blood type heterozygous B, there's only 1. Making your phenotypic ratio the same with your genotypic ratio. 1:1:1:1. Thank you very much for watching this video, and I do hope that this helped you a lot, especially in guiding you to answer your Science 9 learning activity sheets. If you still have concerns and questions about your topic, about this topic, you can contact me through my contact number and my messenger account. Thank you very much and good day.